So this is the activity for the Chapter 10 in-class worksheet, and we're going to go over the answers. So we're going to start with Part 1, Case 1, Muscular Relaxation During Surgery. Prior to intubation for a surgical procedure, the anesthesiologist administered a single dose of the neuromuscular blocking agent succinylcholine to a 23-year-old female to provide muscular relaxation during surgery and to facilitate the insertion of the endotracheal tube. Following this, the inhalation anesthetic was administered and the surgical procedure completed. So basically, in order to get the breathing tube down your throat during a surgery, they have to give you an injection of a muscle relaxant, and it's called succinylcholine, and so we're going to see how this works to paralyze and relax your muscles. So number 1, beginning with the action potential arriving at the motor neuron synapse, describe the normal sequence of events at the neuromuscular junction which led to muscle contraction. Use the images below to help you describe these events. So you have the images on your worksheet, and we're going to walk through the different events. Again, don't get hung up on the step numbers and what action belongs to what step number. Just make sure you know the series of events and the order in which they occur. So let's, and a brief note here. When you're studying the events of the neuromuscular junction, keep in mind that it starts with an action potential that arrives at the synaptic terminal of the neuron, and it ends with the action potential being generated along the plasma membrane or sarcolemma of the muscle fiber. So we're going from an action potential in a neuron to an action potential in a muscle cell, and that is the key goal of all of these events that happen at the NMJ. So let's set up what actually is taking place here at the neuromuscular junction. So first we're looking at the synaptic terminal of our motor neuron shown in blue. And remember, there's a thin synaptic cleft separating the motor neuron from the muscle itself. And the area of the skeletal muscle cell that the neuron is going to talk to is called the motor end plate. So inside the synaptic terminal, we're going to have vesicles full of a neurotransmitter called acetylcholine. We also have some voltage-gated calcium channels embedded in the membrane of the synaptic terminal. There are some ligand-gated cation channels that can bind to acetylcholine, and these are embedded in the membrane of the motor end plate on the muscle cell. And then finally, we have acetylcholinesterase, which is an enzyme that breaks down acetylcholine, and it's hanging out, just floating around inside the synaptic cleft. So this is our setup. So we've labeled all of the parts that are going to play a role. So the actual process begins when we have an action potential that arrives along the membrane of the motor neuron. So it started up in an area of the axon called the axon hillock, and we'll go over that in Chapter 12. And then it propagated down the entire length of the axon, and now it has arrived at the synaptic terminal. So that action potential has just arrived at the synaptic terminal. That's where we left off. So the action potential is going to trigger the voltage-gated calcium channels to open. And we'll again cover a little bit more detail in Chapter 12, but a voltage-gated channel opens and closes in response to membrane potentials. So the action potential changes the potential of the membrane, and that causes these channels to open. And then because calcium is higher outside the cell than it is inside the cytoplasm, so there's our voltage-gated calcium channels open. So the action potential was a trigger, and then these channels open. And then, as I mentioned, calcium ions are higher outside the cell than they are in the cytoplasm. So as soon as the channel opens, you get diffusion, and the calcium ions are going to diffuse in to the synaptic terminal because they are moving from an area of high to an area of low. So the calcium ions will rush into the cell, and they actually move pretty quickly because it's a very steep concentration gradient. And if you remember from Chapter 3, the steeper the concentration gradient, the faster diffusion occurs. So we've just had the calcium ions come into the cell. So those calcium ions are going to float over and they're going to attach to receptors on the vesicles that contain the neurotransmitter. And when the calcium ions attach to these proteins on the vesicles, they trigger the excitosis of the vesicles. So the influx of calcium into the synaptic terminal triggers the excitosis of those vesicles containing acetylcholine. So as part of the process of exocytosis, which again we covered in chapter 3, the vesicles fuse with the plasma membrane, right? And then they're going to release the acetylcholine into the synaptic cleft. So as they fuse, what is inside the vesicle gets put on the outside of the cell. So now acetylcholine is dumped into the synaptic cleft. Note that at this point, by the time we've gotten down to this stage, that action potential is gone. It's very brief. Action potentials last just milliseconds. They are very short, very brief. So the action potential is gone, which means the voltage-gated calcium channels have already closed. So no more calcium is coming in. And in fact, there are calcium pumps that immediately start to put the calcium back on the outside of the cell. So when you have one action potential, you may only get one, maybe two vesicles that get released. And so this allows other vesicles to hang around and wait for additional action potentials that may come a little bit later. So we've just released acetylcholine into the cleft. So we move on to the next step. So the acetylcholine, then once it's in the cleft, now it can bind to those ligand-gated ion channels that are on the motor end plate, which is the part of the skeletal muscle cell that talks to the neuron. So acetylcholine binds to the ligand-gated cation channels on the motor end plate. The binding of the ACH to these channels causes them to open. So notice that now these little channels are all open. And these are cation channels, which means they let both sodium and potassium pass through, but we're focusing only on sodium for now because that is the key ion that ends up causing the action potential. So even though there is some movement of potassium out of the cell, it's very small. So the net movement is that sodium ions rush into the muscle cell. So remember, as always, sodium is higher outside a cell than it is inside. So if we open up a sodium channel of any kind, sodium is going to rush into the cell. So there is a net rush of sodium ions that come into the muscle cell. And then sodium ions, we're adding positive charges to the inside of the cell. So remember, the inside of the cell had a negative membrane potential. We're adding positive charges. So we're making the membrane potential more positive, and that is called depolarization. So we get a depolarization of the muscle cell membrane, which again is called the sarcolemma. So that depolarization of the sarcolemma turns into an action potential, and that action potential is going to propagate or move along the membrane of the sarcolemma. So we've shown it here as the two lightning bolts that are moving off down the membrane. So those are eventually going to travel down the T-tubules because the T-tubules are part of the membrane of the muscle cell that dives down inside the cell. So we're going to pick that up in the next step. But the depolarization causes the action potential to move along the sarcolemma in both directions. Essentially, that is the end of the events that happen at the neuromuscular junction, but we also talk about what happens afterward to make sure the signal gets turned off. So while this action potential is traveling down the sarcolemma, acetylcholinesterase that was in the synaptic cleft is breaking down the acetylcholine. And because we're breaking down the acetylcholine, those channels close because they require acetylcholine to keep them open. So without acetylcholine to open them, the channels close. Sodium stops rushing in. And so you get that one action potential, but you don't get a second action potential because you have sodium-potassium pumps that immediately start pumping that sodium back outside. So remember back in chapter 3, I told you those sodium-potassium pumps are very important. And that's because anytime sodium rushes into a cell, it immediately gets pumped back out to maintain that concentration gradient. Okay, number 2, what prevents acetylcholine from accumulating in the neuromuscular junction and causing a sustained contraction in a normal individual? So remember, we have that enzyme acetylcholinesterase in the synaptic cleft. As soon as acetylcholine is released by the synaptic terminal, the acetylcholinesterase immediately starts breaking it down to turn off the response. So this ensures that the signal from the neuron is very brief and that the muscle cell is only activated for a brief period of time. We're talking about milliseconds. If you did need a sustained contraction, like for example, you're going to hold something up with your arm and you need your muscle to stay contracted, then the neuron would have to send additional action potentials to release more acetylcholine, because that acetylcholine that was released initially is going to quickly get broken down by acetylcholinesterase. So if I want longer than a very short contraction, I have to keep sending action potentials. And that is back when we talked about the different types or frequencies of stimulation. So we talked about wave summation and tetanus. That gets us back to the action potentials, additional action potentials being sent down the neuron. Number 3, succinylcholine acts on the cation receptor channels in the motor end plate. It prevents repolarization after the initial depolarization, which means that the muscle cell cannot receive another signal from the motor neuron. Explain how this would result in paralysis. What is another mechanism within the neuromuscular junction that would cause paralysis? Hint, curare works by this mechanism. So a little background, and again, this is going to be part of chapter 12. A membrane has to repolarize before it can be depolarized a second time and you get another action potential. So when we added all of those sodium ions to the inside of the cell, we raised the membrane potential, but we have to lower it again back to the negative 85 millivolts if we want to get a second action potential. So what succinylcholine does is it holds those ligand-gated cation channels open so they can't close, and this causes a sustained depolarization of the muscle cell, so sodium can continue to rush into the muscle cell. So what happens is you get a single action potential when those channels first open. So the channels open, you get one action potential, which is called a twitch, and then the muscle can't respond to any additional signals until it is able to repolarize, and it can't repolarize until those channels close. So to go back to our image from a couple of slides ago, basically what is happening with this medication is that the drug, succinylcholine, causes these channels to stick in the open position. So sodium can keep coming in and the muscle cell can never reset itself to prepare for another signal from the neuron. So the initial action potential causes calcium release from the sarcoplasmic reticulum, so we'll get a muscle twitch. But then the calcium is pumped back into the SR and the contraction stops. And we cannot get another contraction until these ion channels up here close and the muscle cell can reset to its original resting potential. So the muscle becomes unresponsive and it can't contract farther, and that is paralysis. Other drugs like curare actually block where the ACH binds. So they sit here in the place where acetylcholine normally binds, which means acetylcholine can't get past them to bind itself. And because they block acetylcholine can never bind to those channels to cause them to open. And if the channels never open, you never get an action potential in the muscle cell and thus no contraction. And if you don't have a muscle contraction, that is paralysis. So the bottom line, the take-home message is that repeated rounds of depolarization and action potentials are required for a muscle cell or in a muscle cell in order to get sustained or repeated contractions that you need when you want to move. So you have to be able to open these channels, close them again, reset the muscle cell, open the channels again, close them again, reset the muscle cell, open the channels again, close the channels again, reset the muscle cell. You have to do that cycle over and over again in order to get more than one contraction in a muscle cell. So in the first case, we looked at normal muscular relaxation during surgery. And so now we're going to look at what happens in some individuals that have a genetic mutation in a condition called malignant hyperthermia. So case 2, the next patient came into the operating room for a minor surgery. As the anesthesiologist began to administer the succinylcholine for intubation, the patient went rigid with muscle contractions. The patient's temperature also soared. "Oh no," cried the anesthesiologist, "we've got a case of malignant hyperthermia." With the patient's pulse rate soaring and blood pressure dropping fast, the doctors had to work quickly to reverse the anesthetic. They were successful and the patient survived, although he would not be having his surgery that day. So let's see what this malignant hyperthermia is and why it happens. So first, let's continue with our steps of muscle contraction by describing excitation-contraction coupling. In other words, what are the events that connect the arrival of an action potential from a motor neuron, that's the excitation part, and the actual contraction cycle of the actin and myosin filaments, that is the contraction part. So remember that we finished off the events at the neuromuscular junction with an action potential that was traveling along the muscle cell membrane or sarcolemma. So in order to couple that action potential, which is the excitation part, to the contraction cycle where we get cross bridge formation between actin and myosin, the following things have to happen. The action potential moves along the sarcolemma and down into the T-tubules. The action potential causes voltage-gated calcium channels in the sarcoplasmic reticulum to open, and then calcium rushes out of the sarcoplasmic reticulum and into the sarcoplasm. Remember, calcium levels in cytoplasm or sarcoplasm, which is the cytoplasm of muscle cells, is always low. You have high calcium outside the cells, you have high calcium inside the SR, but in the sarcoplasm itself, levels are low. So calcium will want to go from the high level of calcium inside the SR to the low level outside the SR, so calcium rushes out of the SR. So this is what couples our excitation to our contraction. The lab help link that I posted for the Muscle Physiology PhysioX lesson shows some animations that can help you visualize these processes. It's in the background information section on that link. And I've also created a video capture of just that one section that I'm posting on D2L that you can also watch. Beginning with the arrival of calcium ions to the sarcomeres, describe the steps of the contraction cycle. So the calcium that was released from the SR arrives at the sarcomeres, and it arrives at the zone of overlap where you have thick filaments and thin filaments together. Calcium binds to a molecule called troponin. Troponin, which is also bound to tropomyosin, changes shape after calcium binds to it. This causes the tropomyosin to roll away from the active sites on actin, which now become exposed. Once the active sites on actin are exposed, this starts the cross-bridge cycle, which consists of the following repeated steps. A myosin head, which is currently attached to an ADP, attaches to the active site on actin, forming what is called a cross-bridge, which is a physical connection between the thick and thin filaments. The myosin head releases the ADP, which causes it to pivot towards the M line. This is called the power stroke. And it pulls the thin actin filament along with it. A new ATP molecule binds to the myosin head, which causes it to unbind from the active site on actin, so it lets go of actin. The myosin head breaks that ATP down into an ADP, which gives it the energy to return to its original position, pointing away from the M line. So now we have the myosin head bound to ADP pointing away from the M line, so we can repeat. So now that myosin head attached to the ADP attaches to an active site on another actin, forming a cross bridge, and this whole cycle repeats. So these 4 steps will repeat over and over and over again, as long as there is enough calcium around to bind to troponin to keep tropomyosin rolled away from the active sites on actin. As soon as that calcium is pumped back into the SR, this whole process will stop because the tropomyosin will then roll back to cover the active sites on actin. And again, there is an animation to help you visualize these processes in the link posted and in the separate video that I'm going to post. So number 6, in cases of malignant hyperthermia, a genetic mutation causes the calcium channels in the sarcoplasmic reticulum to react to certain anesthetics by staying open longer, releasing much more calcium than normal into the muscle fiber. How does this explain the rigidity seen in these patients? So because those calcium channels stay open for longer than normal, calcium is released from the SR faster than it can be put back in using the calcium pumps. So we have excess calcium that's hanging around in the sarcoplasm. And so because we have that calcium that's still in the sarcoplasm, it still binds to the troponin molecules. So the troponin molecules continue to bind to calcium, and this keeps the tropomyosin continuously rolled away from the active sites on actin. As long as the active sites on actin are exposed, the cross bridges can continue to form between myosin and actin, and the contraction cycle that we just described, cycle continues indefinitely. This results in a sustained muscle contraction, which explains the rigidity seen in the patient. So basically, all of the skeletal muscles in the patient contract and stay contracted. Number 7, when ATP is used for muscle contraction, some of the energy stored in each ATP molecule is released as heat. Review the 6 functions of skeletal muscle and choose the function that is best explained by this phenomenon of heat release. So the 6 functions of skeletal muscle tissue are: it helps to produce skeletal movement, so that's caused by the muscles pulling on your bones. It also helps us maintain our body posture and body position, so even when we're not moving, we can still sit and stand still based on muscles. Muscle tissue also helps to support soft tissues in the body, especially in the abdominopelvic cavity where we don't have any bones to protect those organs. Skeletal muscles guard the entrances and exits into and out of the body. This would be things like the sphincters, like your voluntary anal sphincter or your voluntary urethral sphincter, and even the throat muscles where you swallow liquids and food to get it into your digestive system. Skeletal muscle tissue also helps with maintaining body temperature and with the storage of nutrient reserves, especially amino acids from the muscle proteins and glycogen, which is a stored form of glucose. And then to answer the second question, choose the function that is best explained by this phenomenon of heat release. That would be the maintaining body temperature. So the release of heat as each ATP molecule is broken down into ADP is what helps us to maintain our body temperature. Question 8. Compare and contrast the metabolism in a resting muscle, a moderately active muscle, and a muscle undergoing peak activity. Use the chart below to help you. So let's start with a muscle at rest. So the ATP demand would be very low. So we have a low demand for ATP because we're not contracting, we're not forming cross bridges. So we have more than enough oxygen available to make the ATP we need. And in fact, the mitochondria can not only meet the ATP demand, but they can make extra ATP. The muscle at this point is using aerobic metabolism, so it is using oxygen and the mitochondria, and it is making excess ATP. So because we have a low demand for ATP, the mitochondria can make a lot of extra, which can be stored. And at this point, our energy source is actually fatty acids, and that way we can take any glucose that's hanging around and use that to store as glycogen for the future. So as far as energy reserves, we're taking the excess ATP and we're storing it as creatine phosphate, and we're taking the extra glucose that we're not using because we're using fatty acids and we're storing it as glycogen. So a muscle undergoing moderate activity has an increased demand for ATP. There is enough oxygen available to meet that demand. The mitochondria can meet the demand, but they're not making any extra or any surplus ATP, so they're making just enough to meet the demand. We are still using aerobic metabolism as our main source of ATP. And again, we're making just enough ATP, but no more, no extra, no surplus. The energy source, first the muscle will use up the glycogen, break it down into glucose, and then use that glucose as the primary energy source. But if glycogen stores start to run low, it will switch to using fatty acids. And during moderate activity, the glycogen stores are converted back to glucose to use for the aerobic metabolism. So in a muscle at peak activity, we have an enormous demand for ATP. We don't have enough oxygen around to meet that demand. The mitochondria is working as hard as it can, so it has maxed out activity, but it's still only making about 30% of the ATP needed, so it's still not making enough to meet the demand. So now, to get that other 70% of the ATP, we have to switch to anaerobic metabolism, which is just using glycolysis. It's more inefficient, but it's the only option at this point. So there is a deficit in the amount of ATP that we are producing, so we actually need to go and use our stored energy reserves in the form of creatine phosphate. And the energy source here is going to be the glycogen broken down to get to the glucose, and so it'll be the glucose that'll be used in both the mitochondria and glycolysis. Now we're going to our CP, our creatine phosphate reserves, and we're using that to convert and make more ATP, and we're also converting our glycogen into glucose. So question number 9, using that chart we just filled in, what level of activity is occurring in the patient undergoing malignant hyperthermia? So since all of their skeletal muscles are stuck in the contraction cycle and undergoing continuous contraction, their muscles would be working at peak activity, so the last row in that chart. They are using all of the available oxygen, and then they're still also using anaerobic respiration in their muscles. They will also be using up all of their glycogen and creatine phosphate reserves. Number 10, what does the word hyperthermia mean? What could explain the hyperthermia seen in this patient? So hyperthermia, hyper, remember, means more, like with hypertonic. So hyperthermia is an elevated body temperature. So don't confuse that with hypothermia, which you've probably heard of before. Hypothermia is when you're too cold. That's H-Y-P-O, hypo. With hyperthermia, your body temperature is actually elevated. So a fever is a type of hyperthermia. Since all of the patient's skeletal muscles are contracting continuously and using ATP to do so, they are releasing a lot of extra heat energy. So imagine every muscle in your body contracting all at once. This rapidly increases the body temperature, resulting in hyperthermia. So number 11, malignant hyperthermia is rapidly life-threatening. That's why it's called malignant. The malignant part means life-threatening. Patients must quickly be given a drug called dantrolene. Given what you know so far about what happens in patients with malignant hyperthermia, predict a mechanism by which dantrolene can stop the runaway muscle contractions. So dantrolene has to in some way work to stop the muscle contractions. Now you might have come up with different ideas if you did this worksheet before checking the answers on how we could stop a muscle contraction. But remember that in malignant hyperthermia, the calcium channels that release calcium from the SR are staying open too long, causing too much calcium to be released. So the optimal solution would be if you mentioned something or some way to find a way to close those calcium channels, or maybe even to pump the calcium back into the SR faster, right? Something to do with getting rid of the calcium that is in the sarcoplasm. And in fact, if you want to know what dantrolene actually does, it does bind to the calcium channels in the SR and it prevents them from opening. So those channels can no longer open. So the calcium is no longer released from the SR, and that's why it is used in these cases of malignant hyperthermia. So number 12, as stated in the previous questions, malignant hyperthermia is rapidly life-threatening. Think about possible reasons why. Hint, think about muscle fatigue, resources in the body being used up, and waste products being produced. These changes have sometimes been referred to as a metabolic storm. Now, don't expect you to know the actual answer to this question. This is more of a critical thinking question. So I wanted you to think about ways that this condition could be overall dangerous to the body. So your answers may vary and may not be exactly the same as the ones I'm about to give you, which are the real answers. If you want to check one of your answers to see if you were on the right track, you can email them to me or post them in the D2L discussion boards. So here are some actual examples of why this condition can be fatal. So a high body temperature can injure cells and cause the proteins to denature or unfold, which means they can't do their job. This is actually why a fever that is too high is also life-threatening. So if your fever gets above like 106 degrees Fahrenheit, your doctors will panic. The skeletal muscles are also using up all of the body's oxygen, which is causing the heart to beat faster and the breathing rate to increase, putting more strain on the body. The rapid heart rate, which is called tachycardia, can lead to hypertension, which is high blood pressure, and cardiac arrhythmias. If not treated quickly, the entire cardiovascular system can collapse, which is seen by the sudden low blood pressure that the patient experienced. The skeletal muscles are producing more carbon dioxide as a byproduct of the increased metabolism, and they're also producing lactic acid from the anaerobic respiration. And both the CO2, which you'll learn about in A&P2, and the lactic acid cause the blood to become too acidic, and this sends the body into a condition called acidosis, which you'll also cover in A&P2. And as the muscle fibers become damaged and die, their internal contents get dumped out into the bloodstream, and too much of this all at once can overload the kidneys, which would normally filter these compounds out and cause acute kidney failure. So basically, it's a very bad condition, and it all happens very quickly. And to give you an idea of how serious this is, in the 1970s, if someone had this genetic mutation, often they wouldn't know until they had surgery, and the doctors wouldn't know until the patient suddenly went rigid on the operating table. And the mortality rate was greater than 80%, which means 8 out of every 10 people that this happened to would end up dying on the operating table. However, with current diagnosis and prompt treatment with dantrolene, mortality rate is now down to less than 5%, so you don't have to worry that you might have this mutation that would cause this reaction. Number 13, once a patient recovers from malignant hyperthermia, their skeletal muscles have to undergo a period of recovery. Describe what happens during the recovery phase. So any lactic acid that was released into the blood following the use of the anaerobic respiration is filtered out by the liver and converted back to pyruvate. The pyruvate will be used to make more ATP using aerobic respiration, or it can be converted back to glucose where it came from in the first place and then stored as glycogen for later use. Because the muscle cells would have depleted their stored reserves, they have to replenish them. Lots of oxygen is required to make the ATP needed for this using aerobic respiration. So this is sometimes called settling the oxygen debt. The oxygen is used to make ATP. Some of that ATP is used to make glucose and convert the glucose back to glycogen. ATP is also used to build back up your creatine phosphate or CP reserves. And until your reserves are replenished, your breathing rate and the depth of your breathing will remain elevated. And then the body has to get rid of extra heat as well. Normally, the body will sweat and dilate blood vessels in the skin to get the extra heat away from the body and into the environment. In the case of malignant hyperthermia, the hyperthermia is more extreme than it normally is. So the doctors and nurses in the operating room will help to reduce the body temperature quickly by using chilled IV saline solution and or putting chilled saline directly into the body in a procedure called lavage. In part 2, we're going to look at Duchenne muscular dystrophy, abbreviated DMD. So James Finlow, a 19-year-old male, is immobile and hospitalized for pneumonia. He has a long history of progressive weakening of his muscles. In the first year of his life, James reached many motor skill milestones, such as holding his head up, rolling over, sitting, and standing at the normal times. However, he did not walk until age 16 months, and by age 2 started to assume a lordotic posture, lordosis, while standing but not while sitting. Over the next several years, he suffered progressive muscle weakness, most notably in the proximal musculature of the arms, pelvis, and legs. By age 9, he required braces to assist his walking, and by age 11, he was confined to a wheelchair. In his early teen years, James was still able to use eating utensils, write, and type on a keyboard, though these functions have declined over the past year. At 16, he was hospitalized with bronchitis requiring antibiotic treatment, but recovered. The only medication that he normally takes is calcium supplements. A musculoskeletal exam revealed a scoliotic deformity, scoliosis, of the spine when James sits up. The upper chest, shoulder, and thigh muscles were significantly atrophied, but the calf muscles appeared enlarged. So number one, a biopsy of James's muscle revealed certain microscopic changes to the skeletal muscles. First, the muscle fibers appeared to be degenerating, and there was a proliferation of endomysial connective tissue around the degenerating muscle cells. Describe the normal anatomy of a skeletal muscle, including a description of the 3 layers of supporting connective tissue. At what level are the above described changes taking place? So a skeletal muscle, the organ, is composed of bundles of muscle fibers called fascicles. So the skeletal muscle or organ is the largest structure, and it's made up of these bundles called fascicles, and inside the fascicles is where you find the individual muscle cells or fibers. So if we look at the 3 supporting connective tissue layers, the epimysium is the outermost layer that surrounds the entire skeletal muscle organ. The perimysium is the middle layer that surrounds the fascicles, which are those bundles of muscle fibers. This layer contains the blood vessels and nerves. And then the endomysium is the innermost layer that surrounds each individual muscle cell or fiber. This layer also contains capillaries, which are the smallest type of blood vessels, and the synaptic terminals of the neurons. This layer also contains myosatellite cells that allow limited repair of muscle tissue. So the last question said at what level are the above described changes taking place? Notice that it meant that the muscle fibers or cells were degenerating and there was a proliferation of endomysial connective tissue. So that means that in this disease, changes are taking place in the individual muscle cells and the surrounding endomysium. Basically, what is happening is that the muscle cells are dying and the endomysium is taking their place. Also, just remember that the collagen fibers from all 3 of these layers, epimysium, perimysium, and endomysium, come together to form the tendon or aponeurosis. Question 2. Describe the internal anatomy of a single skeletal muscle, which is a single cell. Use the picture below to help you. So first in this picture, we can identify the sarcolemma, which is the plasma membrane of a muscle cell. We can also see inside the cell, which is the sarcoplasm, so that's the same thing as a cytoplasm in a normal cell. We also see quite a few mitochondria because cells have a very high demand for ATP. And then the bulk of the muscle cell is made up of these bundles of myofibrils. The myofibrils are made up of the myofilaments, which are the actin filaments and the myosin filaments, so thin and thick filaments. So now we can label, now that we're looking at a myofibril that's been stretched out here, we can see that we have the thin filaments that are made up of actin, and they're attached to the Z line over here. And then we also have the thick filaments, which are made up of myosin, and they are attached to the M line shown here. And together, the thick and the thin filaments from one Z line to another, which is hidden, makes up one sarcomere. And so the myofibrils are repeating units of sarcomeres. We can also see the T-tubules or transverse tubules. These are enfoldings of the sarcolemma. So you can tell in this picture the sarcolemma actually goes inside the cell to form these little tunnels. So the T-tubules are basically extensions of the sarcolemma that go inside the cell. We also have the sarcoplasmic reticulum, which is abbreviated SR. It is similar to the smooth ER you find in a normal cell, except this one only stores calcium ions. And we can also see a region of the SR over here called the terminal cisternae, which is the swollen ends of the SR. And finally, we can see a triad, which is made up of one T-tubule and then the 2 terminal cisternae on either side of it. Number 3, in Duchenne muscular dystrophy, there is a genetic mutation in a gene that codes for an important membrane protein found in muscle cells. As these muscle cells deteriorate in James, extracellular calcium can enter the cell through holes in the sarcolemma. What effect do you think this has on the muscle cells as they deteriorate? So I just included this picture just if you're curious. Muscular dystrophy, the mutation is in a protein called dystrophin. That's where the name dystrophy comes from. And dystrophin is part of the anchoring proteins that anchor the sarcolemma. So in these particular patients who have this mutation, this protein is abnormal, and so it causes weakening of the sarcolemma or the plasma membrane, and it allows calcium ions to be able to cross to get into the cell. So remember that calcium levels are normally low inside the cell in the sarcoplasm. So calcium is either kept outside of the cell in the extracellular space, or if it's inside the cell, it's being kept inside the SR, the sarcoplasmic reticulum. But the levels actually floating around in the sarcoplasm are normally very, very low. So with Duchenne muscular dystrophy, extracellular calcium can enter the cell because of the deterioration of the sarcolemma. And when that calcium comes into the cell, it can bind to troponin. So normally the calcium that binds to troponin comes from the SR, but any calcium that leaks into the cell from the outside can also bind to troponin. And that causes tropomyosin to roll away from the active sites on actin. This allows cross bridges to form between actin and myosin, and that leads to a muscle contraction. Since the calcium continues to leak into the cell, even as it's being pumped back out or pumped into the SR, the contractions continue. Basically, you cannot stop the calcium link because of the mutation that has messed with the dystrophin protein. So, as the muscle deteriorates, it gets locked into a permanently contracted state. So number 4, as muscle cells degenerate, the tightness that results in certain motor units are called contractures. What is a motor unit? Another microscopic feature of these muscle cells is extensive overlap between the myosin and actin filaments. What does this mean for the muscle's ability to produce tension? In other words, discuss the relationship between sarcomere length and tension production. So first I wanted to show you a picture of what we mean by contractures. So remember, we said in the previous slide that as the muscle cells deteriorate, calcium is leaking in, and that is causing the muscles to permanently contract. And so in different motor units throughout the body, in these patients, you see these permanent contractions, and they can distort the limbs of the body into odd shapes and positions, like you see with the hands and the feet in these images. Eventually, the muscle cells will die, but in the process of them dying, you get these contractures. So to answer the question, a motor unit is all of the individual muscle fibers controlled by a single motor neuron. And then if we talk about the relationship between sarcomere length and tension production, keep in mind that the amount of tension that a muscle can produce is dependent on the number of cross bridges that can be formed between the myosin and actin and how much room the sarcomeres have to shorten. If there is too much overlap, so the sarcomere is already shortened, then you can form cross bridges, but there's no more room to shorten, and so no tension is produced. If there is too little overlap, so the sarcomere is stretched, then not enough cross bridges can form, and so you still don't get any tension. Therefore, the maximum amount of tension is produced when the zone of overlap is large, so lots of cross bridges can form. But you still have plenty of room between the ends of the thick filaments and the Z-line so that you can get shortening of the overall sarcomere. So that's why we use this graph called a length tension relationship graph, and it's usually always in a bell curve shape. And so if your sarcomere is too short, you have too much overlap, so you can't shorten anymore. If it's too long, you don't have any overlap, so you can't get cross bridges, so you don't get any tension. And then right in the middle is your Goldilocks area of optimal overlap, where you get the highest amount of tension or force. So that's where you have optimal overlap. You can get lots of cross bridges and you still have room to shorten. Okay, number 5. In patients with Duchenne muscular dystrophy, many of the skeletal muscles atrophy while the calf muscles typically enlarge, hypertrophy. What is the difference between atrophy and hypertrophy? Note, in this disease, it is actually pseudo-hypertrophy because while the muscle enlarges, it is because the muscle tissue is being replaced with collagen and adipose tissue. Bonus question, look ahead to chapter 11. What are the two major calf muscles called? So atrophy occurs when a muscle cell or an overall muscle is not being used. There's a reduction in the muscle size, which reduces its overall muscle tone and its ability to produce tension. Atrophy is reversible until the muscle fibers die. Once they've died, there's no bringing them back and it becomes permanent. So in muscular dystrophy, the muscle cells die eventually, and that results in irreversible atrophy. Hypertrophy is the enlargement of the overall muscle due to the production of more mitochondria and more myofibrils and larger reserves of glycogen. So in Duchenne muscular dystrophy, the muscle cells in some areas, for example, the calf muscles, get replaced by adipose tissue. It is still unknown why this happens in some areas and not all areas, but it makes the muscles look larger, but it's not actually true muscle enlargement. It's just that the muscle got replaced with adipose tissue, and that is why it's called pseudo-hypertrophy. Pseudo means it's false. So it looks like hypertrophy, but it's not really. And so here's a picture of someone in early stage of Duchenne muscular dystrophy. So you can see that they have these enlarged looking calf muscles, although muscles in the other areas of the body become smaller and weaker. And to answer the last question, the two major muscles of the calf are the soleus and the gastrocnemius. Number 6. At the time of diagnosis, a blood test revealed that James had elevated serum creatine kinase levels. This is the enzyme that can add and remove a phosphate to creatine. What is creatine, and how does a muscle cell use this molecule? What do you think elevated levels of CK in the blood might mean? Hint, this is normally an enzyme found inside muscle cells. So creatine is a molecule that muscle cells use to store extra energy. When you have excess energy, phosphate groups from the ATP are transferred to the creatine to make creatine phosphate. So here's the formula. ATP plus creatine gives you ADP and creatine phosphate. And if we look at it in a picture form, we have ATP and creatine. We take one of the phosphates from ATP, we stick it onto the creatine to make creatine phosphate, and then that leaves us with ADP, and CK is the enzyme that does this. When you have a deficit of energy, the creatine phosphate can transfer the phosphate back to ADP to make more ATP. So the formula would be creatine phosphate plus ADP gives you ATP plus creatine. And again, if we look at it in picture form, we take the creatine phosphate and an ADP. Creatine gives the phosphate back to make ATP, and then because it doesn't have a phosphate anymore, now it's just creatine. And again, CK is the enzyme that also catalyzes this reaction. So to answer the last part of the question, CK, creatine kinase, is normally found inside muscle cells. So elevated levels of CK in the blood is an indication of muscle cell death. As muscle cells die, this enzyme is released into the tissues where it eventually makes its way into the blood. So this is actually one of the ways that muscular dystrophy is diagnosed. Number 7, review from previous chapters, because we love to go back and remember and connect the dots. What is lordosis and scoliosis? Why do you think James suffers from these conditions? How does this relate to the functions of skeletal muscle? So lordosis is an exaggerated lumbar curve, also called swayback. And if you look at the second image here, so this would be a really deep curve in your lower back region. That is called lordosis. So very early on, patients with Duchenne muscular dystrophy show lordosis when they are standing. And then scoliosis is an abnormal lateral curvature of the spine. So as the patient becomes older and they become wheelchair bound, and you can actually start to see the spine curving left to right like it's not supposed to. So lateral curvature. One of the functions of skeletal muscle is to maintain body posture and position. So as James's skeletal muscles atrophy and degenerate, especially the muscles in the trunk, they can no longer support the vertebral column, and this results in abnormal spinal curvatures, and they end up with both lordosis and scoliosis. Number 8, why is it important for James to take calcium supplements? Hint, it might not be related to the muscle itself. So I know we've been talking a lot about calcium. We've been talking about calcium in the synaptic terminals. We've been talking about calcium in the SR in the muscle cell. But don't forget that we also have calcium levels in our blood, and the bones also use calcium. So James is wheelchair bound. Also, as his muscles atrophy and die, he becomes unable to put weight-bearing stresses on his bones. So due to the lack of the weight-bearing stresses, the osteoclast activity in the bones would increase, and James would start losing bone mass. So taking calcium supplements can help to counteract the bone loss from the inactivity and the lack of exercise. So by now you can probably see just how important calcium is to a lot of physiological processes in the body. And this is why we introduced you to calcium regulation in Chapter 6, and we're going to talk about it again in A&P2, because calcium is a very critical ion for a lot of physiological processes. So is sodium. Number 9, critical thinking. Why is James susceptible to respiratory tract infections? So the muscles that allow us to breathe, like the diaphragm and the external intercostal muscles, those are the two major ones. There are others. These are all skeletal muscles. They have to undergo cycles of contraction and relaxation in order for us to breathe. So they contract when we inhale and they relax when we exhale. You'll cover more of that in A&P2. But as James's skeletal muscle deteriorates, it becomes more difficult for him to breathe. So his breaths become more shallow, so he's not getting air as deep into his lungs. It would also be difficult for him to cough. Coughing and sneezing also require skeletal muscle contractions. And therefore, because he can't move as much air in and out of his lungs and he can't cough to help move mucus, he starts getting mucus accumulating in his lungs, and mucus is a great food source for bacteria. So this provides the perfect condition for bacteria to invade and infect his lungs. So he gets repeated respiratory tract infections like bronchitis and pneumonia. And then just for your information, since we are talking about Duchenne muscular dystrophy, and you might be curious by now, DMD is typically fatal by the ages of 25 to 26. It used to be rare to live to the age of 30. Now, because of modern medical treatments, you can maybe sometimes make it to 40, but still you're not going to have a very long life. The primary cause of death is usually finally respiratory failure and the inability to breathe. There is currently no cure, but lots of research. And it primarily affects males because it is an X-linked mutation. That means the mutation that causes it is on the X chromosome. So because females have 2 X chromosomes, if we get a normal copy and a bad copy, we're still okay. But a man only has an X and a Y, so if he gets a bad copy on his single X, he has the disease. So this is why it primarily affects males. And that is the end of the Chapter 10 activity.